I would prefer to be there in person, but glad that we have the opportunity to communicate via this forum. Today I'll be talking about neurocorrelates and mechanisms underlying treatments for addictions. As I do these days, I have a listing of disclosures with respect to pharmaceutical, gambling, gaming, and legal relationships. And I thought I would start by talking a bit about what we mean by addiction, because the use of the term has changed over the course of the past several millennia. Initially, the word is derived from a word meaning bound to or enslaved by and was not linked to substance use behaviors. Then it became increasingly linked to substance use behaviors. But I would argue that over the first course of this millennium, there's been a swinging of the pendulum back such that behaviors like gambling and gaming may be considered as addictive. So with respect to treatment development for addictions, there have been both successes and areas where we still need to improve. And for some addictive disorders relating to cannabis, cocaine, gambling, gaming, there are no medications with indications for these conditions. And we have been interested in understanding mechanisms underlying therapies, both behavioral therapies and pharmacological therapies, particularly at a brain-based level, so that we can improve and optimize treatments and targeting of treatments. So we've proposed multiple different targets with respect to neurotransmitter systems that might be considered both for behavioral and pharmacological treatments for substance addictions. And one major model or several models are dual process models that suggest that there are reward-related motivations that might drive engagement in addictive behaviors in a bottom-up fashion, whereas executive control processes exert top-down control over engagement in the behaviors. And we and others have used two tasks, the Stroop color word interference task, which is a cognitive control task, and a reward processing task, the monetary incentive delay task, in some of our studies. We've also proposed that these may link particularly to specific treatments, with cognitive control being particularly relevant to cognitive behavioral therapy, and how people process monetary rewards being particularly relevant to contingency management, for example. And one widely used task that we have been using, as I mentioned, is the monetary incentive delay task. And this task was developed several decades ago based on work by Wolfram Schultz in non-human primates. And some of the early studies in humans found that individuals with alcohol dependence showed a blunted activation of the ventral striatum, a brain region that had long been implicated in reward processing, in anticipation of working for monetary reward. Since that time, this finding has been extended to individuals at risk for addiction, such as those family history positive versus negative for alcohol use disorder or alcohol dependence, as well as for individuals with tobacco use, gambling, and other disorders. And in cases where it's been examined, there's been found to be an inverse relationship between ventral striatal activation and clinically relevant measures like impulsivity. So these are data from a meta-analysis that includes some of our studies that show that individuals with substance use disorders and gambling disorders are characterized by similar blunted striatal activations during reward anticipation on the monetary incentive delay task. So a major effort of our group has been to incorporate these fMRI measures into randomized clinical trials. And we have been interested in understanding the brain mechanisms underlying the proposed active ingredients of therapies. Some of our early studies looked at brain activations prior to treatment and related them to treatment outcome. So here, this is a study of 20 individuals and with cocaine use disorder. And what we found was that brain activations during Stroop performance linked prospectively to treatment outcome measures, both self-reported abstinence and urine toxicology-based abstinence. And these implicated brain regions such as the striatum and medial prefrontal cortex and cingulate cortex. Retention was also found to be related to brain activations during Stroop performance, and this implicated more dorsolateral prefrontal cortex. We also have used a variety of different analytic approaches to try to understand these relationships. And so the prior slide used a whole brain analysis using a general linear model-based analysis, but some other alternate approaches like independent component analysis may identify functional networks linked to important processes. Whereas others, like Connectome-Based predictive modeling, may identify how different brain networks interact, and intrinsic connectivity distribution is another functional connectivity-based approach that is a data-driven approach. So we applied independent component analysis to those 20 individuals who I presented earlier, and also looked at between group differences. And we found that there were 6 components that were identified that distinguished the cocaine individuals with cocaine use disorder from those without, and those are shown here. And some of these, like the singular opercular and singular motor functionally integrated activations or brain networks, distinguished individuals with and without cocaine use disorder, whereas the medial singular frontal circuitry was related to retention, and largely frontal cortical, frontal subcortical brain regions were implicated in abstinence from cocaine. So this is providing some insight at a circuit-based level into brain activation patterns linked to treatment outcome. So as I mentioned, intrinsic connectivity distribution is a data-driven approach that is a threshold-free approach, and this, we believe, helps to avoid certain inconsistencies and findings. And when looking at a group with cocaine use disorder and those without, we found that during Stroop performance, there was greater functional connectivity with subcortical brain regions and frontal brain regions that have largely been linked to hot processes. So these included regions like the striatum, including ventral components, and the orbital frontal cortex. And some of the subcortical brain regions, ventral striatum, thalamus, insula as well, were linked to positive cocaine urine toxicologies. Now, we've also looked at how brain changes during the Stroop performance change from before treatment to after treatment. And what we found in our initial studies of 12 substance-using individuals was that there was a relative decrease in brain activation following treatment as compared to prior to treatment that was over and above a test-retest group. And we have subsequently replicated that finding in a group of individuals with cocaine use disorder in a randomized clinical trial involving cognitive behavioral therapy, contingency management, and disulfiram. And both cortical and subcortical brain regions implicated in cognitive control were identified. And these findings together suggest that perhaps there's greater efficiency of brain circuitry function after treatment when individuals with cocaine use disorder are exerting cognitive control. Now, we saw a very different pattern of brain changes when we looked at monetary incentive delay task responding. Specifically, as one can see in the top set of slices, there is greater activation after treatment as compared to before treatment in regions implicated in reward processing, including the ventral striatum and insula, as well as amygdala and inferior frontal gyrus. Now, we've also utilized Connectome-Based predictive modeling to understand brain network changes that occur with treatment. And we found that both positive and negative networks could predict abstinence, and there was a significant relationship between predicted and actual abstinence in the functioning of the models. And these networks involved a broad range of brain regions that can be then segregated into different networks. And these plots show relationship between different networks. So for example, this hot plot here for the positive network between the frontal parietal and medial frontal regions suggests that these are more functionally integrated in predicting cocaine abstinence, whereas some other brain networks like medial frontal and salience network are more segregated. So if we take these together, this allows us to show proposed models where there's between network integration and between network segregation that is linked to drug abstinence, in this case, cocaine abstinence. In the United States and elsewhere around the world, there are opioid epidemics, and we had questions about whether similar brain network patterns would be linked to opioid abstinence. We used Connectome-Based predictive modeling on a similar sample as a lot of the individuals with cocaine use disorder were in methadone maintenance treatment. And what we found was that there was a different network that predicted opioid abstinence. And this involves stronger within-network motory-sensory connectivity and reduced connectivity between motory-sensory networks and the medial frontal, default mode, and frontal parietal networks. So these findings suggest that there are distinct networks related to outcomes in specific addictive disorders. Taking those findings and breaking them down a bit, the full Connectome-Based predictive modeling worked best for cocaine abstinence using the Stroop data and for opioid abstinence using the monetary incentive delay task data. But once those networks were identified and applied, one can see that both resting state and the alternate tasks fit the abstinence models, but the opioid models did not fit the cocaine data, and the cocaine data did not fit the opioid data. And additionally, another point of showing the robustness of the data is that if we virtually lesioned a network, the remaining network remains significantly contributing to the model, suggesting a robustness of the findings. Now, white matter integrity is another brain measure that we have collected in our studies of treatment outcomes for substance use disorders. And what we found is that poorer white matter integrity is associated with shorter duration of cocaine abstinence in individuals with cocaine use disorder. We've also collected volumetric measures and have found that the relationship between pre-treatment cocaine use and treatment outcome with respect to urine toxicologies is a significant relationship that appears to be fully mediated through hippocampal volume. Now, we've also been interested in understanding adolescents versus adults, as there are data to suggest that brain regions mature, making adolescence a period of addiction vulnerability. And part of that relates to how the frontal cortical regions mature less rapidly in adolescence as compared to subcortical brain regions. And some of our data from adolescents and adults, when they are performing a similar task involving personalized cues related to stress or favorite food conditions, indicates that the adults show largely cortical brain responses and adolescents largely subcortical brain responses. So this raises questions about adolescents and their behaviors and engagement in addictive behaviors like smoking. And what we found was that when we had adolescent smokers performing the Stroop, they showed largely similar brain activations during task performance as do adults. And when we looked at relationships of pre-treatment brain activations to treatment outcomes, we observed that some similar brain regions, including medial prefrontal cingulate regions, were implicated. Similarly, in adolescents, as in adults, post-treatment brain activations during Stroop performance were less robust than pre-treatment on the Stroop, but on the monetary incentive delay task, as in the adults with cocaine use disorder, post-treatment measures in the ventral striatum were more robust than they were at pre-treatment in adolescent smokers. Now, I had mentioned previously the meta-analytic findings suggesting a blunting of ventral striatal activation during monetary reward processing in addictive disorders, including substance use and gambling disorders. Some of our data using a reward processing task suggests that something similar might be happening in internet gaming disorder. And what we found in adults with versus without internet gaming disorder is that there is less functional coherence of an executive control network involving frontal parietal brain regions in adults with internet gaming disorder as compared to those without. And the degree of functional connectivity within this network is linked to out of the magnet cognitive control as assessed during Stroop performance. Similarly, in a meta-analysis, we observed that individuals with internet gaming disorder show less gray matter volume in brain regions that have been implicated in inhibitory control, such as the anterior cingulate gyrus, as well as those implicated in motivational drive, such as the orbital frontal cortex. And based on these and other data, we've proposed mechanisms underlying internet gaming disorder and how therapies might target specific aspects of these domains. We have also tested, in conjunction with groups in China, Chentao Zhang and colleagues at Beijing Normal University, we've tested a craving behavioral intervention in internet gaming disorder, observing that the active intervention as compared to a test-retest group was associated with decreases in internet addiction severity, weekly gaming time, and craving responses to gaming clips. Furthermore, the active intervention as compared to test-retest was associated with a greater response of the anterior insula to the gaming clips following the intervention. And this, if we use that brain region as a seed, it showed decreased functional connectivity after the intervention as compared to previously in the group receiving the craving behavioral intervention. Also, we collected resting state functional data and found that the group with and without internet gaming disorder was distinguished bifunctional connectivity differences involving the orbital frontal cortex and posterior cingulate. And following treatment, there was relatively decreased functional connectivity of these regions to regions implicated in memory processing as well as motor preparation and enactment, suggesting a possible mechanism by which the craving behavioral intervention might be operating. So other groups, such as that led by Klaus Wölfling, have tested cognitive behavioral interventions for the treatment of internet and computer game addiction, and have found it to be efficacious. And we've also considered non-substance use disorders like binge eating disorder, but we've also considered them within an addiction framework, considering relationships with the proposed entity of food addiction, citing both similarities and differences between binge eating disorder and substance use disorders. And when we look at how individuals with binge eating disorder perform on the monetary delay task, we see some similarities with what we have observed and what others have observed with individuals with addictive disorders. That is, a blunted activation in the ventral striatum in the individuals with binge eating disorder and obesity as compared to those with obesity alone. Furthermore, there's individual variation within the group with binge eating disorder such that those who have the more blunted activation of the ventral striatum at treatment onset do worse during treatment for binge eating disorder with respect to showing persistent binging. So taken together, the findings suggest that while there are many treatments for individuals with addiction, there are specific disorders for which we do not have therapies, and perhaps by integrating brain imaging measures into randomized clinical trials, we can get a better understanding of the brain mechanisms underlying these disorders and their treatment, and that this may help us target interventions through novel approaches such as neuromodulation, and we think that it will be important to consider both the diagnostic entities as well as intermediate phenotypes that may be clinically relevant as we try to optimize treatments for people with addictions. So I'd like to thank a large number of individuals and a number of funding sources, and thank you for your attention.